Hello, everyone. This is Bill Harris, together with my daughter, Dr. Jackson, here to talk about the strength study. We thought this would benefit from a rather thorough discussion, and so we hope this is helpful to you. We've entitled this Strength, What Happened? And we'll get, if you don't know what strength is, you'll find out in just a moment. I am with the Fatty Acid Research Institute and the University of South Dakota School of Medicine. Kristina is also with the School of Medicine and at Omega Quant Analytics, Director of Research there. So let's get into this. So new findings, big headlines, well, at least in the medical world. Strength trial questions cardiovascular benefit of high-dose omega-3, and then strength trial finds new fish oil medication did not reduce risk. So this is what the strength study is. This was reported from the American Heart Association in the middle of November 2020. So let's talk about this study. So I'm going to keep moving this thing around. Let's see, put it down there. So the strength study, the title of it was the effect of high-dose omega-3 fatty acids versus corn oil on major adverse cardiovascular events in patients at high cardiovascular risk. It was a parallel arm, placebo-controlled, double-blind trial, very standard drug model for doing a study. Patients were high risk for cardiovascular disease. They had to have elevated triglycerides and low HDL cholesterol, and a variety of other risk factors that we'll see in a moment.
So that's more of a cardiometabolic risk person.
Yeah, right, exactly.
More obesity and some insulin resistance and some heart disease and
blood pressure.
Yeah, metabolic syndrome.
Yeah, 13,000 people, so it's a big study, 22 countries. The treatment was 4 grams a day of Epinova, which is a free fatty acid form of EPA and DHA. They're called omega-3 carboxylic acids, which is simply the same thing as saying if it's a free fatty acid. But they're referred to as omega-3 CAs, carboxylic acid, instead of EE ethyl ester. Total dose a day of EPA DHA was around 3.1 grams, because there's other fatty acids in this product. The placebo here was corn oil, 4 grams a day, so less than about a teaspoon of corn oil. 50% of that is omega-6 linoleic acid. And
just to be clear, the free fatty acid form of omega-3s has never been tested at this dose for this long. Like all the other omega-3 studies, none of them were in this form. Right. Much more typically in ethyl ester. Both Vascepa and Loveza have been studied as ethyl esters. This was about a 3.5-year follow-up study, but about 3 years of exposure to the drug. So that's what we're talking about here. And the outcome, as mentioned, major adverse cardiovascular events, which are listed there. It's all kinds of bad things: death from cardiovascular disease, non-fatal MI, fatal MI. Fatal MI is part of cardiovascular death. Non-fatal stroke, coronary revascularization, meaning angioplasty with a balloon and stent, or unstable angina requiring hospitalization. So if any one of those events happened to a patient in the study, it's considered an event. They had the outcome. Didn't have to have all of them, just one of them is fine. That's the way that composite works. Patient characteristics are shown here. 63 years of age on average, mostly, or 65% were men. BMI was 32, so they were on average obese, over 30. 82% were white, 11% Asian. As mentioned, it was 22 different countries around the world. 56% of them had a prior cardiovascular history, 71% had diabetes, 88% hypertension, 10%, and the background medications. Everybody was on a statin. 71% were taking some kind of a platelet active agent, and 67% were on beta blockers. So that's the kind of patient we're talking about. This is not certainly what you call primary prevention of generally healthy people. Yeah, so everybody in both the group
placebo and treatment group was on that level of background medication the whole time.
The whole time, true, right. So here's the short story, cut to the chase. What do we find? No difference at all. This is the percent of patients with an event on the y-axis and the months after randomization on the x-axis. So you can see that there is no difference between the line for the corneal placebo group and the omega-3 CA, carboxylic acid group on the primary endpoint. So this is what all those headlines were about. There was no effect. So let's think about it. Buried within the paper, there is a few places of statistically significant benefit for omega-3. One of them here is in coronary events. So that's non-strokes, heart events in patients with established cardiovascular disease at baseline. So there was a significant difference here. 0.85 means 15% reduced risk, p-value of 0.02. So that was significant. But by and large, all the outcomes they looked at in all the subgroups, largely no effect of the treatment, except for this particular one. If you look for good news, you've also got to look for bad news. And probably the worst news from this, if anything, would be the increased risk for atrial fibrillation in the omega-3 group. And again, it's shown here. It basically went from about 2% of patients in the omega-3 group, about 1.3% of patients in the corneal group developed atrial fibrillation during the study. And that's a significant increase in risk. And as noted here, this agrees with a trend that was seen for higher AFib in the Reduce It trial and in another study we're not going to talk about today. So there is some evidence that something about these high dose omega-3s and AFib needs to be explored further. Side effects are important to look at, and it may be important, particularly in this study. And what we have here is the gastrointestinal. There were 25% of the people in the omega-3 group that had reported gastrointestinal problems, compared to about 15% in the corneal group. Diarrhea, nausea, the biggest one is diarrhea. And so the question does come up, was the drug difficult to tolerate? Perhaps not from an omega-3 point of view, but from a formulation-free fatty acid point of view. Importantly, there's no increased risk of bleeding.
So in other studies, do you recall there being a difference in the GI adverse events in omega-3 groups?
Yeah, but they're always much smaller than this.
Mm-hmm.
And it's usually, a burp, fishy burp. That's the kind of thing that they call an adverse event.
Yeah.
So this is a little higher than expected in other trials.
Yeah.
So omega-3 blood levels are reported in this study. And this table shows what they were. Here's the EPA and here's the DHA level in both the plasma and the red blood cell. Actually, the red blood cell should be as a percent, not microgram per mil, so the units are not quite right here. But if we actually take these data and estimate what the omega-3 index is, we would come up with 5.6% would be the baseline level in both the corneal and omega-3 groups, which is very typical of at least Americans. Of course, this is worldwide, but it's very typical omega-3 level. And it really didn't change, dropped a little bit maybe in the corneal group, but it went up to about 9.4% in the omega-3 group, which is a good change, an expected change. We like to see the omega-3 index of over 8%, but we'd like it to be there more than for 3 years to see benefit. We think that's a lifetime thing.
And
comments on that?
The reason why they were testing, or one of the reasons to test the free fatty acid formulation, is because they'd seen in other studies that it was very bioavailable. Is that right?
Yeah, it increased omega-3 levels in the blood quite readily. It doesn't really require enzymatic breakdown.
to get absorbed like an ethyl ester does.
Mhm.
So that was the presumed strength of, pardon me, strength, is to use a free fatty acid form.
Yeah.
So, anyway, they got in the blood. That's the point
Yeah.
here.
Yeah.
Now they have a table in the paper where they try to ask the question, but they don't do it very well. Was there a difference in outcomes as a function of the change in omega-3 levels in the blood? And they looked at it in a, they calculated a percent change in omega-3 level for each person. And then they put those percent changes into tertiles. And the people with the biggest percent change were compared to the people with the least percent change. And they asked if there was a relationship with that across those 3 tertiles with outcomes, and they didn't find anything. We think that's a problematic way to do that. You want to explain your thinking down here at the bottom, Tina?
Yeah. So these are percent change from baseline. And it's number one, it's just confusing even for us to look at this, and we look at blood level stuff all the time. So that's not the best. As you can see, this example of why only looking at percent changes can be a little misleading is, especially with with EPA here, RBC EPA as an example, if you went from 0.3, which was which I think it was like the first quartile or tertile level of EPA, and then you go up to 1.65, that is a percent change of 450%, which is that highest change group. But even when you have that big of a change, you still aren't at even the mean level of EPA for the treatment group. So you might have had a really big change percent wise, but your blood level is really not that high. So it's a little bit tricky to understand this. And also we had talked about before, if somebody, baseline levels are part of a percent change calculation, but they're not quite controlled for here. So what we would like to see is, number one, just look at what blood level people got to by the 1 year mark. And so you could do a tertile analysis, same idea, but just have, split into low, medium, high final blood levels. We would think of it in the omega-3 index terms of what the 3 different levels of omega-3 index. And then you would pool everyone together. You would not continue to split out between treatment group and placebo, because there's a possibility that people in the treatment group didn't change hardly at all. And there's a possibility that people in the corn oil group changed a little bit more than they should have. So you just look at blood levels at 1 year. Then we also would like to see not just looking at tertiles, but actual forcing people into groups. So it wouldn't be equal across the ends would not be equal across the groups. But if we looked at people who had an omega-3 index less than 4 and greater than 8, which are kind of our cutoffs that we've been using here, and omega quant, look and see what the event rates are between those groups. And then finally, changing it up a little bit and using baseline omega-3 index levels or EP and DHA levels, and splitting up the tertiles that way. And then you maintain that treatment and placebo group comparison. So then you can see what you would hope to see is those people with the lowest omega-3 index at baseline. That's where the difference between treatment and placebo might show up. And then as you have higher and higher baseline levels, you see less and less of a treatment effect. And that kind of analysis is done in a lot of other studies that I've seen.
So this is not in this paper, and something we would hope to be able to maybe work together with AstraZeneca to look at these kind of outcomes.
Mm-hmm.
Good. Okay. So to sum up, large, well-run trial, different kind of omega-3, again, the free fatty acid form at a high dose, roughly 3 grams a day. And they did measure endpoints before and after. They didn't recruit people based on a low omega-3, but they at least measured blood levels, which is a step in the right direction. And they, of course, followed for hard cardiovascular endpoints, meaning very clearly distinguished endpoints.
And more than blood levels, actually.
What?
More than blood levels of like cholesterol or
yeah, right.
Like a heart attack. That's something very real.
That's hard, right. Again, they added omega-3s to standard of care regimen, which is several drugs for most of these people. And these folks were pretty high risk to begin with. And so, of course, they did not find an effect. We still have questions about what omega-3 levels were achieved and how, if there's thresholds of protection, if there's a risk plateau based on blood levels, et cetera, like we mentioned before. So that's the sum up of strength. Now, I think, Tina, you want to take over from here?
Sure. So this strength study comes out, and as you'll see, it's very similar in design and study population to the Reduce It trial. And so it made people start looking at the Reduce It trial again. There was already some speculation here about the placebo in that study, but it kind of reopened this this discussion. So we're going to kind of show you how much these studies mirrored each other and look at what potentially happened. You go on. So this is the design of the Reduce It trial. It's the same, basically the same thing as the strength trial. They had a placebo, it was 2 arms and double-blinded, same type of population, a little bit smaller, but still a worldwide study with 11 countries involved. 4,000. The big difference is the active ingredient, the EP. It's just EPA and it's ethyl ester. And so those two differences between the treatment groups and then the placebos were also completely different. The strengthened was corn oil, and the placebo here is mineral oil at 4 grams a day. And that is really, the mineral oil is the big question mark, I think. 5 years of follow-up here and the same criteria for endpoints. And here you can see the populations are almost identical. Same type of population. We do see when we look at concentrations of EPA, because Reduce It was an EPA only drug, they only reported EPA and they only reported plasma concentrations. So in strength, they reported that as well. And you can see the strength EPA level did not get as high on average as the Reduce It level. So that could be important.
Yeah.
And then here is a laid over graph. Because these populations and these endpoints were almost identical, and they both reported the MACE event rate, we thought, while we were looking at it, we were noticing just like the actual event rates in strength were lower than what they saw in Reduce It. Because my other question is, what happens to what's the event rate for a normal population? What if, the omega-6 and the omega-3 in strength are both potentially beneficial? That's a question. We don't have that information. So we can compare it to another study, and this one is as close as you're going to get study-wise, population-wise and design-wise. And what we see is that the 2 lines at the bottom are from the strength study. And you can see as you get to about one year, you have about 5% of people in both groups having events, or maybe 4. And then year 2, it's about 7, 3, it's about 10, 4, it's about 15, maybe a little less. And then you can see in Reduce It, both the placebo and the active group are above those thresholds, above those lines. So there were more people in that study that were having events.
Here's the 25% reduction in risk that they reported and Reduce It.
Yeah. And so if you have the argument when Reduce It came out, because the results were so good, was that this placebo is unusual and also potentially increasing risk. So then your treatment is actually just more like normal, and your placebo is actually doing the bad thing. Instead of, usually we expect a placebo to be, this is normal, and our treatment should be improving things.
Right.
And that's
that's a very informative graph. Has to be taken with a little grain of salt, but it does raise the question, as others have raised, is that maybe in Reduce It, the apparent benefit of EPA was not really a benefit of EPA, but a increase in risk, partly from the mineral oil use. I think mineral oil has never been used for 4 or 5 years as a daily dose of anything. Typically, it's used in large doses for a week for constipation, but it's not used at like a teaspoon a day every day for 5 years. So who knows what effect that has on the absorption of, let's say, fat-soluble vitamins that might not be absorbed very well. Maybe it affects the absorption of lipid-soluble drugs. Some statins are lipid-soluble, and maybe that prevented some of that absorption. We still can't tell, but it raises a question of why the placebo group in strength had such a lower event rate than the placebo group in reduce it.
Mm-hmm. Yeah, because you always have to have the context of like the study didn't work. It didn't work compared to placebo.
Right.
And there's also just the effect of being in a study. And that's why you have a placebo group, is like when you're in a study, you're probably taking all of your meds more regularly, and you're probably, so that's why you have your placebo. But then, yeah, what happened here? Yeah, we can't say for sure, but that's what the rates looked like. So yeah, a lot of the mineral oil stuff had been argued when Reduce It came out, and the point about statins maybe not being absorbed as well on the mineral oil group. And do you want to kind of explain these numbers?
Sure, yeah. It was noticed earlier on that in the placebo group in Reduce It, LDL cholesterol went up. It was statistically significant. It wasn't huge, from 76 to 84. Not the kind of rise that you'd think would be particularly problematic. CRP levels went from 2.2 on average to 2.8. Okay, again, it's an increase, yes. Statistically significant, yes. Clinically significant, unclear. Of course, these are only 2 metrics that they measured that gave some fuel to the fire that the placebo in Reduce It was not really inactive. It was actually harmful. We still can't know for sure. Did corn oil reduce risk and strength? This is another suggestion that corn oil is, omega-6 is a beneficial cardioprotective fatty acid, but given it, again, 4 grams a day, which is 2 grams of linoleic acid, that's quite a small dose. Hard to imagine it really had any important effect, but it's conceivable. It didn't change LDL cholesterol levels. CRP levels went down, if anything. They went down with the omega-3 too.
Mm-hmm.
Other suggestions people have raised is maybe DHA in the strength trial, which was absent from the Reduce It trial. Maybe it neutralized the effect of EPA. There's no evidence for that, other than speculation.
But the pure EPA had the Jealous trial was also successful on the just EPA ethyl esters
at 1.8 grams
at a lower dose, and in Japan in a higher background omega-3 diet.
Right.
And so maybe there's something.
And there was no placebo in that trial.
Right.
So there was no mineral oil for sure.
Mm-hmm.
No placebo. And so they found a benefit there. That would argue that the EPA only is active and beneficial. So that's an argument in that favor. Also, it could be that in the strength trial, which again used free fatty acids, as opposed to the ethyl esters, there may have been some kind of irritation of free acids in the stomach or the small intestine that caused a kind of a chronic inflammation to go on in strength that eliminated some of the apparent benefit of omega-3. Would not have been an omega-3 problem, would have been a drug formulation problem.
Right.
But
that's where also the CRP numbers for strength are not complete. They only have about 3,000 analyzed here instead of 13,000. So they reported on a small proportion of total. So you get all the CRP numbers, you might be able to tease out. I mean, it probably is like a subgroup of people who are having.
hopefully a representative random subgroup.
Yeah, hopefully.
So,
So what now?
yeah, what do we do?
So then there's the omega-3s don't work crowd with all these studies. I think it just, omega-3s are blessed and cursed because they are seen as both a drug and a nutrient because of the pretty powerful nutritional studies. We kept pushing them more into the drug realm, and a drug is it's a very specific population that these are tested in. It's on top of a bunch of other drugs, and it's in people who are already sick. And a nutrient is supposed to be for a general population, getting enough nutrition or of these different types of nutrients throughout your lifespan so you don't get certain illnesses. I mean, the classic is the nutrient deficiencies, but also we think about in chronic disease, having proper nutritional status is important to prevent having chronic disease. So omega-3s might not be good drugs. I mean, in this paradigm, it's pretty inconsistent. The EPA ethyl esters are the only ones who have had any success, and those trials are being reassessed and kind of looked at again. And it's not perfect, but nothing is. But does that mean that omega-3s are not an important nutrient? I don't think so, but I'm biased.
And a large reason there from the beginning why they were called drugs was so that the companies that manufactured them, sold them, could get reimbursed through medical systems for the costs, because they're expensive products. And if they're not drugs, they're not reimbursable. Mm-hmm. So they were, first of all, approved as treatments for very high triglycerides. And that made it an FDA officially approved drug for a particular outcome, high triglycerides. And then doctors were able to prescribe it, and they were reimbursed. Patients were reimbursed for that. Mm-hmm. Doctor was. So that's how they became drugs. Again, it's a molecule. It's the same molecule as now EPA ethyl esters and Lovesa. Ethyl esters are not natural forms, but as soon as you take them and digest them, they get in the blood just like a natural form does. Mm-hmm. So drug nutrient, it's kind of a false dichotomy in a way.
Mm-hmm. But I think how we think about it and how we test how drugs work versus how nutrients work, it's just
right. Windows too short.
So then that begs the question, how do we do research moving forward? Because I'm definitely interested in seeing how we can break away a little bit from this just clear placebo treatment group. You give everyone in the treatment group the same dose, regardless of their baseline status, and it doesn't matter what number they get to, it's just you were given this dose. I want to see studies that are analyzed based on blood levels that people achieve. Or I want to see titration studies where they test people's levels over the study. And once people get to that level and maintain it, then we start to look at outcomes. That's something I think in the omega-3 space is very easily done. We're talking to some people who are trying to start it, but it's a really different way to do a study than the normal two groups. And it will probably take a while for that kind of work to get recognition or acceptance, because it's not the gold standard. And then the other issue is just the clinical utility of doing that in the clinic, in the real world. We typically work with doctors that are more in the prevention realm than the treatment realm, I would say. And so they're more looking at getting people's blood levels up earlier on in a disease process than potentially a cardiologist that is seeing someone because they're having heart issues. So using it, having optimized omega-3 status to prevent things like I talked about nutrients, I think is a different idea than what is being tested in these large drug studies.
Right. And that's a good wrap up. I think Thanks for joining me on this and putting together this little talk. And if anybody has questions, certainly contact Kristina at Omega Quant or me at the Fatty Acid Research Institute. Happy to respond and discuss this further.